Hello and welcome back. Today we're going to pick up with viruses and how they fit into our study of genes and genetic expression. And viruses are kind of an interesting study because they're not really alive. They're included in biology because they require a living cell as a host, but they're microscopic infectious agents that reproduce inside host cells. It comes from the Latin for slimy liquids or poison. And the curious thing is they exhibit some but not all of the characteristics of life. And you can see from these two diagrams here, here is an adenovirus, a computer model of that. And then over here is an image from the 1918 Spanish flu outbreak, where 500 million people worldwide, about a third of the population, were infected, and between 50 and 100 million were believed to have been killed by the virus. So what are viruses? Well, they're not living by themselves, but they can reproduce at a fantastic rate, and this leads to mutations, and this has given them an incredible number of viruses worldwide. Number two, they are acellular. So they're not cells, they don't have their own metabolism, and they're dependent upon host cells. That means that they're obligate parasites. They can't reproduce unless they infect and take over a host cell's machinery. And finally, one thing they have in common is they all contain some type of nucleic acid, and that can be single-stranded DNA, double-stranded DNA, single-stranded RNA, or double-stranded RNA. And if you look at this diagram here, this will give you some examples. The top row here, here's the ones for double-stranded DNA. So herpes virus, the pox viridae, smallpox, cowpox, and then there's also single-stranded DNA. Now the bottom ones are ones that have RNA as their nucleic acid, and here we can see the retroviruses, the orthomyxoviridae, and some others, coronaviridae, which is SARS you may have heard of, and the common cold. So first of all, the size is they're microscopic. They're incredibly small compared to what we've been looking at so far. So if you look at this diagram with the animal cell and its nucleus in the background, and even the bacteria, the virus itself is much smaller than half a micrometer across. So we're talking about an incredibly small organism or agent here. So the structure of a virus comes in two parts. So there's an inner core that's made of nucleic acid, and as we said before, this is either DNA or RNA. The RNA, many of those will be what we call retroviruses, which we'll talk about in a later edition. And then second, they all have a protective outer coat of protein called the capsid or the capsule. So if you look at this diagram here, here's three examples of that protein capsid or capsule on the outside with the nucleic acid in the middle. So there's polyhedral. Here's one that's helical. So again, the capsid is almost in scales on the outside with the nucleic acid. And here's one we call a complex shape, or a bacteriophage, where the polyhedral capsule on the outside and the DNA would be in the middle. So those are the two parts. So it's a very simple structure. And then the shapes, and we're going to concentrate on four main ones. So the first is there's the polyhedral, which is the adenovirus, which causes upper respiratory tract infections, is an example. And here we've got a polyhedral protein capsule around the outside with the nucleic acid in the middle. And there's two kind of cartoon representations at the bottom. The second shape is rod-shaped or helical, and so here's the rhabdovirus, which is rabies, and the tobacco mosaic virus, and these ones are kind of rod-shaped. They've got a protein coat on the outside with the DNA or RNA down the middle. The third one is the bacteriophage, and these are specialized because they infect bacteria, and they've got a polyhedral head with a hollow sheath between them, and so the DNA is in that capsule, and it's almost like a syringe. When they infect the bacteria cell, they inject their DNA right into the host, and the legs here have receptors they'll recognize on the cell wall that allow them to attach and infect that cell. And the fourth one we'll talk about is called an enveloped virus. And these have a coating of phospholipids and glycoproteins derived from the host cell membranes. So this is how they kind of sneak their way into some cells. So they get those receptor molecules on the outside as they leave the cell in a process called budding that we'll look at the very end of this presentation. So HIV, influenza, the mumps and measles are examples of enveloped viruses. And here's a diagram that shows all four of those together. So over on the left here, we've got the rod-shaped. Then we've got the polyhedral, the adenoviruses. Influenza would be an enveloped virus. And then finally, we've got the bacteriophage as we go through for this one. And then we've also got the kind of nice ones. We've got the rhinovirus, the toga virus, and the ever-popular retrovirus that we will talk about. HIV as an example, and we'll look at the life cycle of that. And so what we're going to do right now is we're going to look at, as we wrap up for this one, is how those enveloped viruses are packaged and released by the cell.
Enveloped viruses are usually released from the host cell by a budding mechanism. First, viral spike proteins are inserted into the host cell membrane. Next, the inside of the host cell membrane becomes coated with viral matrix protein. The viral capsid then becomes completely enclosed by the region of the cell membrane into which the spikes and matrix protein are embedded, and the virus is released by budding.